Yes. Ladies and gentlemen, good evening. I'm Martin Rees, the President of the Royal Society, and it's my great pleasure to welcome everyone here tonight. Most people here, I suspect, have been to the Royal Society before and know a bit about it. But for those who haven't, let me just mention that we are the Scientific Academy for the UK and the Commonwealth and have been such since the year 1660. This evening, we are having a lecture which is the last in our series of 6 lectures supported by the Templeton Foundation. And I will say a few words about this in a moment, but before that, I need to give the usual routine announcement. Please turn off your mobile phones completely. And I say that especially because not only do we have the audience in this room, we have an overflow audience in another room, and we also are web streaming this lecture live, so there will be people watching it from all over the world. And also after the lecture, it will appear archived on our website. So for all those reasons, please keep your mobile phones turned off. Well, turning now to this evening's lecture, R Kandel has had a very distinguished career researching the mechanisms of the brain, particularly memory, and he continues this work to this day as Kavli Professor and University Professor at Columbia University and Senior Investigator at the Howard Hughes Medical Institute. He received the Nobel Prize in Physiology or Medicine in the year 2000 and, of course, has received many other prizes. He has many friends in this country. I'm glad to see a number of them here tonight. And he visits quite frequently. He was here last time, in fact, at the Society when his wonderful book, In Search of Memory, was shortlisted for the 2007 Royal Society Prize for Science Books. We're delighted to welcome Eric back to the Society, and it's my great pleasure and privilege to invite him to give his lecture, which is entitled, We Are What We Remember, Memory and the Biological Basis of Individuality. R Kandel.
Thank you, Sir Martin, for your very gracious introduction. I'm really delighted to be here for several reasons. Sir Martin, in an unusual moment, was unusually modest. This is not simply the Academy of Britain. This is the greatest scientific academy in the world, and it's my privilege to be here. I'm particularly delighted to see not only colleagues, but friends and relatives in the audience, and it warms the cockles of my heart to see so many of you here. In my talk tonight, I would like to discuss memory storage and disorders of memory storage. Since this may be the first time that some of you are exposed to research on higher mental processes, I also want to use this opportunity to give you a sense of how brain scientists go about relating mental function to brain function, and having done so, how scientists then go on to analyze brain function on a molecular level so as to try to develop therapeutic approaches that can reverse mental disorders. Let me begin by putting some of the recent progress in memory research into perspective for you. In the last 5 decades, the 5 decades of my scientific career, we've witnessed a remarkable increase in the explanatory power and range of biology that is having a broad impact on all aspects of modern thought, including how we think about the mind and therefore how we think about ourselves. As a result, when intellectual historians look back on this period, they are likely to acknowledge that the deepest insights into the nature of mental processes will not have come from the disciplines traditionally concerned with mind. They will not have come from philosophy, from the arts, or even from psychology or psychoanalysis, but from biology. This is because in the last 2 decades, biology has participated not simply in one, but in 2 major unifications of thought which bear on our understanding of mind. First, there's been a remarkable unification within biology itself. This has brought together into a common molecular science the various subdisciplines of biology: cell biology, developmental biology, immunology, the biology of cancer, and even the biology of nerve cells, the building blocks of the brain. Second, there's been a parallel unification between neuroscience, the science of the brain, and cognitive psychology, the science of the mind. This second unification is far less mature than that brought about by molecular biology, but is potentially equally profound. For it has already provided us with a new framework for understanding perception, action, and memory storage. These two independent unifications stand at the extremes of the biological sciences, and they raise the question, to what degree can these two disparate strands be brought together? Can molecular biology, which provided the driving force for the unification of the biological sciences, enlighten the study of mental processes? Can we anticipate an even broader synthesis in the 21st century, a synthesis ranging from molecules to mind, a synthesis that would open up a new molecular therapeutics? In my talk this evening, I would like to outline the possibility of a new science of mind, a molecular biology of cognition, and suggest that it will occupy the center stage in the early part of the 21st century, much as the biology of the gene occupied center stage in the last half of the 20th century. I outline this development by using as an example the study of memory. I focus on memory because this is my area of competence. The study of memory has occupied my own research interests over these last 50 years. My aim over this time has been to try to develop a reductionist approach to learning and memory that might allow me to explore the underlying molecular mechanisms. Learning, as you well know, is the process whereby we acquire new information about the world, and memory is the process whereby we retain that knowledge over time. Learning and memory have proven to be endlessly fascinating mental processes, and many of the people here have contributed fundamentally to this new science of mind with their fascination of mental processes. And we find it fascinating because it addresses one of the most remarkable aspects of human behavior, our ability to acquire new ideas from experience. Most of the ideas we have of the world and our civilization we have learned, so that in good measure we are who we are because of what we learn and what we remember. Conversely, many psychological and emotional problems are thought to result, at least in part, from experience, and specific disorders of learning and disturbances of memory haunt the developing infant as well as the mature adult. Down syndrome, the normal weakening of memory with age, and the devastation of Alzheimer's disease are only the more familiar examples of a large number of diseases that affect memory. My purpose in attempting a reductionist approach to learning was not to replace psychological thinking with the logic of molecular biology, but to contribute to a new synthesis, a new science of mind that would do justice to the interplay between the mentalistic psychology of memory storage and the molecular biology of cell signaling. This synthesis has recently begun to take shape, in good part because of a number of the people here, and it is these beginnings that I will try to outline for you. It is convenient to divide the study of memory into two parts, the systems problem of memory and the molecular problem of memory. In the systems problem of memory, we ask, where in the brain is memory stored? In the molecular problem, we ask, what are the mechanisms whereby that storage occurs? Let me begin with the systems problem of memory. The history of trying to localize memory storage in the brain really is part of a much larger saga, a much larger exploration, in which scientists ask the question, can any mental process be localized to any region of the brain? And one of the great intellectual accomplishments of brain science in the 19th century was the beginning exploration of mental process localization within the brain. This began in Vienna, where I was born, but earlier than I was born, with the work of Franz Joseph Gall, a remarkable man who made two extraordinary contributions to brain sciences, which is still fundamental to our thinking. Gall insisted that all mental functions derive from the brain. He did away with all dualistic thinking. There was no soul. What was important was the fact that all mental functions derive from the brain, number one. Number two, he said, mental functions are not unitary, and the brain is not unitary. different mental functions are localized to different regions of the brain. And the outer part of your brain is called the cerebral cortex. And he said the cerebral cortex is not a single organ. It consists of many organs, 40-odd organs, and each organ is concerned with a different mental function. He localized them in very specific ways, giving each region a specific mental ascription, sublimity, cautiousness, and he divided this into groups. The intellectual functions he localized to the front of the brain. The instinctual functions, such as cautiousness, amativeness, romantic love, parental love, the back of the brain. And the sentiments in the middle of the brain. This is sublimity, hope, veneration, virtue, all the things that this society stands for. This was an extraordinary set of insights, and they've persisted, if you will, in principle to this day. What was flawed with Gall's approach was the method that he used for localization. He had a complete disdain for routine experiments. He had a disdain for using clinical material to gain insights into localization. He did not want to study patients with strokes or tumors who had disturbances of mental function. He wanted to study the normal brain, and he developed a specific notion which allowed him, according to his own rules, to localize mental function. He argued that as you use a particular mental function, the area that represents that function bulks up, just like a muscle would bulk up as a result of exercise. As it bulks up and gets larger in mass, it pushes on the skull overlying that particular region. And so if you go around and just feel people's brains and see the skull, and I look at John O'Keefe there, I can tell frontal area, intellectual, very well developed. You can just tell by feeling their skull or even just observing it from a distance. I won't even comment on Richard Morris. So this was really quite remarkable. And the way he got this idea was pretty much the way I'm getting this idea. He looked at the colleagues that he had, and he saw that the profound thinkers, those capable of making subtle distinctions, had the front of the brain quite prominent brow, while people who were shallow, among his acquaintances, had, very undistinguished brows. He extended this to all areas of knowledge. For example, he was struck with the fact that people he thought were extremely good mothers had prominent backs of the head, while those that were poor mothers had a very shallow back of the head. And so he argued that obviously this is due to the fact that the area concerned with parental love expanded in the extremely good mother. And the Frigidaire mothers, of course, didn't have this kind of expansion. This made an enormous impact in the scientific community. It was particularly popular in the United States. I'm sure you can understand why. A good story sells very well in the United States. And there were people started societies of phrenology devoted to feeling each other's heads. Often they didn't stop with the head, but they felt each other's skull. They felt each other's skull in order to ascertain the character structure of the person underneath. But not everybody was convinced. The French, as usual, were skeptical. And Marie Jean-Pierre Florence, an experimental neurologist, didn't believe in this theorizing. He wanted to do specific experiments. So he tested the idea that this area in the back of the brain, which Gall attributed to romantic love, amativeness, he wanted to see, is it actually involved in romantic love? So he removed this area in a number of experimental animals, like this, and he found that it interfered with certain aspects of motor function, but in no way interfered with their capability for making love. So he reached the conclusion that what Gall said is not at all correct. He did not think there was localization of function to the cerebral cortex. He thought the whole cerebral cortex functioned as a single organ. And he said, and I quote him in translation, "All perceptions, All volitions occupy the same seat in these cerebral organs. The faculty of perceiving, of conceiving, of willing merely constitutes, therefore, a faculty which is essentially one. This made an enormous impact. People really began to appreciate the fact that Gall, although he may have had a good idea, they no longer had confidence in the idea because the experiments underlying it were very, very poor. In fact, they were really not based on good experiments at all. But the situation changed dramatically in 1861 with the work of a remarkable guy, Pierre-Paul Broca. Broca was an extraordinary neurologist. He was also an anthropologist who was very much influenced by Gall. He thought there's likely to be localization of function, but he thought you can't get at it by feeling the skull. You have to actually get into the brain itself. And he said, I had the thought that if there were a phrenological science, a science of localization, a science for localizing mental functions in the brain, it would have to be a phrenology of convolutions. That is, the indentations and the curvatures of the brain, of the cerebral cortex itself, and not the phrenology of bumps on the head. So you had to get into the brain itself to figure out what was going on. And the way to do that is to correlate clinical symptomatology that you see in a patient with actual lesions in the brain. Clinical pathological correlations. And he had an extremely interesting opportunity to do this. A patient by the name of Laborg presented himself to Broca, and Laborg suffered from an aphasia. Aphasias are disorders of language, and I should tell you at the outset, we have learned an enormous amount in the 19th century from the analysis of people who have one or another disorder of language. Laborg had a particularly unusual disorder of language. He could understand language perfectly well, but he could not articulate language. He could not speak fluently. He could say a few words, but he couldn't form coherent, fluent sentences. Now, you might think this is a paralysis of the vocal cords. It wasn't that at all. He could whistle perfectly well. Moreover, he also could not write language, even though his hand was not paralyzed. So he had difficulty in expressing language. When Laborg died and came to autopsy, Broca noticed that he had a major lesion in the front of the brain. Broca went on to collect a number of patients, and as he studied them, he realized this is an area that seems to be involved in all of these cases. I have to give it a name. So what was the poor boy to do? In all modesty, he named it after himself, Broca's area. Moreover, it struck him that as he examined these 8 patients who had this lesion, that they all had it on the left hemisphere. And this caused him to enunciate one of the great principles of modern neurology. Nous parlons avec l'hémisphère gauche, he said. We speak with our left hemisphere. This made an enormous impact. People really stood up and paid attention. 2 German scientists, Fritz and Hitzig, began to stimulate the surface of the cortex in experimental animals and found that there is a strip of cortex, which we now know is the motor cortex, the precentral gyrus, in which if you stimulate different parts of this precentral gyrus, you would get movements of different parts of the body. So if you stimulate it laterally, you get twitching of the face. If you stimulate it a little bit more medially, you get movements of the arm. If you stimulate it even more medially, you had movements of the leg. Notice that there is a topological representation, a map of the body surface on the motor system. I'm later going to return to this because there's a similar map in the sensory system. So systematically, your body is represented on the surface of the brain so that different parts of that representation are responsible for movements of different parts of your body. A few years thereafter, Carl Wernicke discovered another case of aphasia, a language difficulty, that was the mirror image of what Broca had discovered. Broca discovered a patient who could understand but could not express speech. Wernicke discovered a patient who could express speech but couldn't understand it. When he died and came to autopsy, Wernicke realized that the lesion was quite different Broca's. Broca's was in the front of the brain. This lesion, in which the patient had difficulty understanding language, was in the back of the brain. So again, in modesty, he named it after himself, Wernicke's area. Wernicke, 3 or 4 years out of medical school, made this remarkable discovery, again, on the left side of the brain. Was in an unusual position. He had been second in delineating the language system. Broca preceded him. But I think a wonderful lesson of science is to appreciate the fact that sometimes being second is good because you have the additional knowledge that came with the initial discovery. So he said to himself, look, isn't it interesting? First of all, this shows that Gall is wrong in thinking about how the brain works. A complex function, like language, is not mediated by a single area in the brain. It involves 2 separate areas at least, one for understanding language and one for expressing it. Moreover, he realized that there is a pathway called the arcuate fasciculus, which connects the two. And that allowed him to put the facts together in order to formulate a model of language. Highly simplistic. We now have a much more elaborate one. But this simple model still today explains 85% of what you see clinically in patients with aphasia. He said, look, my area, Wernicke's area, which is involved in the perception of language, is located in the back of the brain. This is where sensory information comes in. This is the visual cortex, in which information from seeing, like reading a book, comes in. Here is the primary auditory cortex, in which information that you listen to comes in. So if you hear language spoken or you read a book, that information in the back of the brain is focused on Wernicke's area, where it gets transformed into a representation of language. That information is then projected on to Broca's area, where the grammar for language is located and where speech is ultimately expressed. And notice he said that Broca's area is located near the motor strip, and that part of the motor strip that is concerned with vocalization. So it's very easy to form proximity. This was not only just an extraordinary model, but it made predictions. In fact, he made a prediction that you would ultimately find patients with aphasia that do not have a lesion in Wernicke's area and do not have a lesion in Broca's area. Can you guess how you can get an aphasia without a lesion in either of those areas? Annie, what's the answer? You disconnect it. This woman is brilliant. She has never gone to medical school. She just figured this out of her own. The disconnection syndrome. These patients understand and they talk, but there's no connection between the two. It's like an American presidential press conference. Information comes in and information comes out, but there's no connection between the two. With this as a background, people began to explore the possibility that maybe memory storage would be localized to some specific region of the brain. Now, this is a subtle question because you could make a number of arguments for thinking that maybe memory storage would not have a discrete site. After all, memory is always associated with something else, either with language or with visual experience. Are there specific regions that uniquely serve to mediate aspects of language function? And the first person to really sort of look at the localization of learning and memory in great detail was Carl Lashley. Carl Lashley worked with rats, and he removed systematically different parts of the cerebral cortex while animals were running complicated maze tasks. And he came to the conclusion that there was no specific area that was concerned with memory storage per se. Rather, that memory storage seemed to require massive amount of cortex. The more cortex you removed, the more you affected memory storage. He developed the idea of equipotentiality, or mass action. And one of the reasons he did this is because he used a very complex task. So if the animal lost one sensation because the visual cortex was damaged, the animal could use smell or proprioception, some other modality to guide it around. The task was so complicated the animal could compensate for it in a number of ways. And so people were skeptical about Lashley's conclusions, which really sounded very much like Ferrand's earlier view. And the person who ultimately addressed himself to this in the most direct way was Wilder Penfield, another really remarkable person. Columbia trained, of course, did not get the chairmanship of neurosurgery at Columbia, so he moved to Montreal, where the Rockefeller Foundation established the Montreal Neurological Institute, the first institute concerned with high cognitive function in people. A student of Sherrington, he developed a procedure that allowed him to work on unanesthetized patients, excuse me, on the surface of the cortex. He realized that the brain doesn't have pain perception. So if you infuse a local anesthetic in the scalp, you can open up the skull and you can stimulate the surface of the cortex without causing any pain. And his specialty was removing scar tissue from the surface of the brain. When people fall and have a head trauma, they sometimes sustain a scar, which can give rise to local epileptic attacks. And his specialty was focal epilepsy, and he cured that by removing the scar tissue in the cerebral cortex that gave rise to it. Now, before you remove scar tissue, you want to make sure you don't damage Broca's area and Wernicke's area. So he would stimulate the surface of the cortex with the patient unanesthetized, so the patient could tell him what he was feeling or what he was experiencing or what he was hearing when a particular region was stimulated. In the course of doing this, he found that in a small number of patients, when he stimulated in this region, the medial temporal lobe, he found memory-like phenomenon. They were sort of like hypnagogic experiences. They were not all that clear. They sort of had a daydream quality to them. Patients remembered vaguely that they were in a room that was familiar to them. They could see a dog chasing a cat. They heard songs that were familiar to them. Penfield was just amazed. This was the only area of the brain that gave rise to that he thought he had localized area of memory storage. Most neuroscientists were very skeptical. They thought that what he was doing is eliciting auras. Patients, before they have seizures, often have sort of hypnagogical phenomenon, like a hallucination, and they thought he was artificially eliciting that. This was not truly related to memory. But that situation changed remarkably a few years later. This was as a result of a very famous patient that many may have heard of, whose name is H.M. H.M., when he was a boy, 12 years old, was run over by somebody riding a bicycle. And he sustained a concussion on both sides of the head, which damaged the temporal lobes on both sides. As a result of this, he had scar tissue on both sides of the temporal lobe, and he had seizures. At first, these seizures were readily controlled. So he was able to continue going to school. He was able to go to high school. He was able to get himself a job in an assembly plant later. But with time, the seizures became worse and worse. And ultimately, he felt he had to do something about it. So he went to Scoville. He lived in New Haven. Bill Scoville was a very good neurosurgeon, trained by Penfield, and asked him, because he knew Scoville was doing operations like this, whether or not he would be suitable for an operation. And Scoville examined him, and since his seizures were so intractable, he decided to operate on him. And he removed the temporal lobe on both sides, including the hippocampus, with a structure deep to it, sitting on the subiculum, the little shelf right here. As a result of this operation, H.M. was cured, essentially, from the seizures. He has still alive today. He has an occasional seizure, and he's on medication. They're very mild seizures. Essentially, the epilepsy was dramatically improved. However, as a result of this operation, he was left with the most amazing memory loss you could possibly imagine, a dense amnesia. Scoville was besides himself. He called up Penfield, and he said, "My God, look what I've done here. I've produced this extraordinary memory deficit in this patient." So Penfield commiserated with him, but he said, "Look, I have a very gifted colleague, Brenda Milner, who British-born, living in Canada, who has been studying people with temporal lobes and has noticed, as have we, that memory is involved with the temporal lobes. She should come down to New Haven and study this patient. And Brenda went to New Haven, studied HM very, very thoroughly, not only on this first time, but over a period of 20 years. And in a brilliant psychological analysis, she described what we now know about the medial temporal lobe and memory. So it was clear that HM had a severe amnesia. He had a severe memory deficit. But what she found was how selective the memory deficit was. There were lots of things that HM could do, even though there was an important thing he could not do. For example, HM remembered almost everything that happened to him prior to the operation. So he remembered the trauma of childhood. He remembered what it was like growing up, going to elementary school. His language was intact. His IQ was in no way changed. So all the knowledge that he had, including his work in the assembly pen, was completely intact. And from this we now know that long-term memory, memories that you've stored for many, many years, do not reside in the hippocampus. They are thought to reside in the cerebral cortex, in different parts of the cerebral cortex, probably those regions that initially process that information. HM also had quite good short-term memory. If you gave him a name, he could repeat it. If you gave him a telephone number, he could repeat it. What HM lacked, to the most remarkable degree, however, was the ability to take that new short-term information and put it into new long-term information. So if you introduced him to Annie, he would say hello to Annie. You introduced him to Elon, he would greet Elon. Come back to Annie, he would have forgotten her name completely. He would sit down with the newspaper. As soon as he read the first paragraph, he forgot it. He would start all over again. Sit down with a meal. As soon as he finished the meal, he forgot he ate it. He would start all over again. Could not remember anything. After a while, if he would look at a picture of himself after the operation, a picture that was 5 or 6 years old, he wouldn't recognize himself. He didn't realize how much he changed. He had forgotten how much he had changed. Every event was a transient event. He could not take any new information and put it into new long-term memory store, despite the fact that everything prior to that, prior to the operation, he remembered perfectly well. So the medial temporal lobe and the hippocampus are involved in taking new information and putting it into new long-term memory. Brenda Milner thought for the longest time that this applied to all areas of knowledge. And then she made another remarkable discovery. She asked HM to draw the outlines of a star, and to do it not simply directly looking at the star, looking at the pencil, looking at his hand, but Leonardo da Vinci-like, much more difficult, looking in the mirror and drawing the outline of the star. Now, when you and I do this, we make a number of errors the first day, but we improve as we try time and time again, 10 trials the first day. Significant improvement. The second day, we pick up, having a substantial improvement, improve a little bit more, and the third day, perfect. No errors whatsoever. When you ask HM, who does just as well as you and I on this task, HM, isn't that great? How come you did so much better on Wednesday than you did on Monday? He says, what are you talking about? I've never done this before in my life. So this is an amazing dissociation. He can learn certain things, but not even remember that he learned them. A number of other people soon made similar discoveries in people who had lesions of the medial temporal lobe. They realized that despite the fact that these people had an enormous memory deficit, that memory deficit was only for certain types of knowledge. Certain motor and perceptual skills they continued to retain. And this gave rise to the view that we now hold, that memory is not a unitary faculty of mind, but has at least two major forms. They're called explicit and declarative, or implicit and procedural, respectively. Explicit declarative memory is what HM has lost. This involves the medial temporal lobe and the hippocampus. And this is a memory of facts and events, a memory for people, objects, and places. So what you normally think of as memory is explicit memory storage. You know, thinking back on your first love experience, thinking back on going to school, thinking what happened yesterday. This is all explicit memory storage. And you have to make a conscious effort. This is one of the defining characteristics, a conscious effort to recall it. But we didn't realize until HM came along and other people like him with dense amnesia came along, that there is in parallel with that a very powerful system, a Freudian system, an unconscious system, whereby we remember skills and habits, and also simple kinds of learning like habituation, sensitization, classical conditioning. And this, in the mammalian brain, involves a number of structures deep in the brain, the amygdala, the cerebellum, and in the simplest cases, the reflex pathways themselves. And what is striking about this is that even though the learning may sometimes require conscious attention, the recall does not require conscious attention. Let me give you an example. When you learn to ride a bicycle, you pay a lot of attention to what you're doing. You tell yourself, now you put your left foot forward, now you put your right foot forward. Once you've mastered that, you don't talk to yourself. If you talk to yourself as you're riding, you're going to fall off the bicycle. When you drive your car and you have to shift up or shift down, you don't tell yourself. You do this automatically. When you rush to the net in order to hit a volley, you don't tell yourself, get your shoulder around. You better do it, otherwise you're not going to get to the ball at all. So these things are reflexly. What is amazing, and this really has emerged as people have studied this to a much larger degree, is how much of life is mediated on this level. I don't know whether any of you have seen the telecast of this British composer, Clyde Waring, who had a dense amnesia. Are you familiar with this? He suffered from herpes simplex encephalitis. He had an unbelievable amnesia. He could remember practically nothing, less than HM. But when you put him in front of his choral group, he conducted as he always did. It was absolutely unbelievable. De Kooning, advanced stage of Alzheimer's disease, could still paint, even though couldn't recognize a person in the room. These are all implicit memory storage. So aspects of creativity in the hands of those who've mastered them becomes implicit. One doesn't even have to call consciousness into play in order to be able to do these things. So this is really a rich category of knowledge that we're dealing here. And it's really very much like Freud pointed out, a great deal of mental life is unconscious, including a significant amount of memory. Now, Freud spoke of several unconscious. The one he's most identified with is the dynamic unconscious. This is not the dynamic unconscious. This is what we would now call his procedural unconscious. The dynamic unconscious deals with sexual conflicts and things like that. But with this as a background, we're now in a position to sort of explore what are the molecules that underlie these two kinds of memory storage. What are the mechanisms that underlie them? So, I mean, as a first approach to it, we can ask, look, these are extremely different memory processes. They use different logic, conscious recall in one case, unconscious recall in the other, completely different anatomical systems, medial temporal lobe on the one hand, the amygdala, cerebellum, reflex pathways in the other. Do they share any features in common? You know, can molecular biology, with its ability to reveal homology relationships, delineate commonalities to these two very different memory processes? Now, it turns out that it was, almost immediately obvious that there are a few things they share in common. To begin with, both implicit and explicit memory storage have a short-term and a long-term form. So there are at least two phases, and there are probably subphases to it. Short-term memory, which lasts minutes, and long-term memory, which lasts days or weeks, number one. Number two, we know how to move information from short-term to long-term. One very effective way is repetition. Practice makes perfect. There are other ways of doing it. If something is dramatically important, one trial would be sufficient to put it into long-term memory storage. But as you probably appreciate from working with your kids and your own experience, repetition helps. And finally, we learned very early on from the pharmacological investigation of Bernie Agranoff, Larry Squire, Sam Barandis, that long-term memory differs from short-term memory in requiring the synthesis of new proteins. So there was reason to believe that on the molecular level, there might be some commonalities. But to really make progress in this, one really has to sort of work in specific cases of memory storage. And I'm going to describe to you studies of explicit memory storage, which are carried in a mouse, and implicit memory storage, which are carried out in the marine snail Aplysia. I should begin my comments by saying that in studies in the mouse, in the mammalian hippocampus, we have here Tim Pliss, Richard Morris, and John O'Keefe, who have pioneered the study of the hippocampus. And I will come to their work as I go along. I didn't know they were coming, but fortunately, I have slides that pertain directly to their work. So if there are any questions, we'll direct it to them. Let me begin with Aplysia. This is a marine snail that my colleagues and I and a number of other people have worked with, and you can see immediately at a glance why you would work with this animal. This is not only a very beautiful animal, this is a highly intelligent animal. Anybody interested in learning and memory would study this animal. This animal, you may not realize it, but it can master classical Latin and Greek. It plays the cello, and the remarkable thing is it has these gifts, and it does so with a nervous system that consists only of 20,000 nerve cells. So your brain and mine has 10 to the 12 neurons, lots and lots of cells. Most of them are really quite small. The brain of the snail is made up of 20,000 neurons. Moreover, these neurons are not randomly distributed. They're collected in clusters called ganglia. Each ganglion, there are about 10 of them, has about 2,000 nerve cells. So that a single ganglion often controls more than one behavior. So the number of cells committed to a single behavioral act can be quite small. And I'm going to describe to you a behavior that involves less than 100 nerve cells. Not only are there few cells, but the cells are gigantic. They're the largest nerve cells in the animal kingdom. Before I became presbyopic, I could see them with my naked eye. So this cell, R2, for example, is a millimeter in diameter. The cells are not only large, but they're characteristic in pigmentation and position, so you can give them names and return to the same cell in every animal of the species. And as a result of this, it's possible to work out a neural circuitry of behavior in terms of specific identifiable cells. And you can look at that neural circuit in a naive animal, an animal that has learned something. In that simple animal with the simple nervous system, we focused on the simplest possible reflex that we could study, the gill withdrawal reflex. This, for the poets in the audience, is the head of the animal. This is the tail. The animal has a respiratory organ called the gill, which is outside the body. It's an external organ. To protect it, there is a sheet of skin called the mantle shelf, which contains the residual shell that these snails have that normally covers it. I've pulled it back so you can see the gill. And that ends in a fleshy spout called the siphon. If you apply a tactile stimulus to the siphon, you get a nice brisk withdrawal of the gill. That reflex can be modified by several different forms of learning, and each case gives rise to a short-term memory and a long-term memory with repetition. I'm going to focus on one kind of learning called sensitization, a form of learned fear in which a noxious stimulus to the animal causes it to enhance its reflex responses in preparation for defense and escape. So if you apply a weak tactile stimulus to the siphon, you get a modest withdrawal of the gill. But if you now give the animal a tail shock, you scare the animal, and now the same stimulus to the siphon produces a much more powerful withdrawal of the gill. The animal remembers this insult, this noxious stimulus, as a function of repetition. You give one training trial, the animal remembers it for minutes. This doesn't require protein synthesis. But you give 5 or more training trials, and the animal remembers it for days or weeks, and this requires new protein synthesis. We worked out the neural circuit of the behavior, and I showed this in very simplified form. There are 24 sensory neurons that pick up from the siphon skin. they make connections to 6 motor neurons that innervate the gill. And the sensory neurons also innervate inhibitory and excitatory interneurons that feed back onto the motor neurons. If you stimulate an isolated piece of skin, you only activate a subpopulation, 4 to 6 sensory neurons. But what one finds is invariably the same sensory neurons always hook up to the same motor neurons, always hook up to the inhibitory and excitatory interneurons. So there is an invariance for the neural architecture of behavior, and that is a general principle. And that posed the initial paradox in the study of learning and memory. How do you get all these capabilities for modifying behavior if the neural architecture itself is so precisely wired? So to explore how this occurs, we looked at the reflex while the animal learned sensitization. And we found when we stimulated the tail, we activated a modulatory system, the most important component of which is serotonergic. And this ended on the sensory neurons, including on its presynaptic terminals, and acted there to strengthen the connections between the sensory neurons and the motor neurons. So what genetic and developmental processes do is to give you the basic architecture of behavior. What they do not specify is the exact strength of these connections. And what learning does is to alter the strength of pre-existing connections. I'm showing you a form of learning that increases synaptic strength. There are some forms of learning that involve a decrease in synaptic strength. If you give one tail shock, you get a transient strengthening that lasts minutes. But if you take the same tail shock and repeat it 5 times, you get a more persistent strengthening that lasts for days, and this requires new protein synthesis. This involves an alteration of gene expression that I'm going to come to in a moment, and a growth of new synaptic connections. So if you remember anything about this lecture in your long-term memory, it's because anatomical changes are occurring in your brain. And I'm going to come back to that. Now, I don't urge this on you. You can leave anytime you want. Let me actually go back 1 second. I'm going to show you a blow-up of the connections between the sensory neuron and the motor neurons to give you an understanding of how this works in the molecular level. This is the sensory neuron, this is the motor neuron, this is the tail, this is the serotonergic system, and this is before we've stimulated the tail. This is in a basal state. An action potential in the sensory neuron will cause a release of a chemical substance we call chemical transmitters, which it carries in these little circular structures, several thousand molecules of transmitter. And that release occurs because an action potential allows calcium, a critical ion, to move into the presynaptic terminal, and calcium is necessary for release. The transmitter here is glutamate, and it acts on AMPA and NMDA receptors. If you now stimulate the tail, you activate these serotonergic neurons, and they act on a receptor that initiates a signaling pathway within the cell. That's called a second messenger system, in which serotonin, the first messenger, triggers an intracellular signaling system within the cell. That intracellular signaling system, in this case, uses a pathway called cyclic AMP and an enzyme it acts on called the cyclic AMP-dependent protein kinase. The details are unimportant. It's sort of the general idea I'd like you to focus on. With a single tail shock, a transient activation of the serotonergic pathway, you only have a modest increase in cyclic AMP. That activates the enzyme only for a short period of time, and it acts here to strengthen the calcium inflow and to strengthen the release of transmitter from the presynaptic terminal. But if you give 4, 5 repeated stimuli, something rather remarkable happens. With repeated stimuli, the level of cyclic AMP goes up more. That allows the enzyme to stay free of its inhibitory constraint and to translocate into the nucleus where the genes are located. In so doing, it recruits another enzyme, a MAP kinase, and they act together to turn on gene expression. And they do this in a very interesting way. They act on an activator of transcription called CREB1, and they have to remove first a repressor, an inhibitory constraint on memory called CREB2. If you don't remove the repressor, you can't activate genes and you can't turn on long-term memory. Now you ask yourself, this is ridiculous. Why would you want to have an inhibitory constraint on long-term memory? Wouldn't you want to put everything into long-term memory? And think about it. You don't want to put everything into long-term memory. Many things that happen to you are miserable. You want to forget them. Most of the lectures you listen to are boring. Who wants to remember this lecture tomorrow? So in order for it to go into long-term memory, it really has to be important for you. This is why repetition and attention are absolutely essential for many kinds of learning. Under those circumstances, the repressor is removed and CREB1 is activated. And it acts on downstream genes to give rise to the growth of new synaptic connections. Now, I really want you to focus in on this because this is an interesting result that many people find surprising. What is a defining feature of long-term memory is the activation of genes. And this is why inhibition of protein synthesis, one of the reasons inhibition of protein synthesis is important, because turning on genes gives rise to messenger RNAs that are translated into proteins. And that's what's required to grow new synaptic connections. But what these studies and other studies have indicated is to turn on a long-term memory, you need to activate genes. Now, many of you probably think of genes as being the controllers of behavior, as the controllers of body form, the controllers of behavior. You must also realize that they're also the servants of the environment. They respond to environmental contingencies, and they alter gene expression as a result of that. Many people get very nervous about this. They see that genes are being turned on as a result of learning. What happens if they go home and they sleep with their partner and they have a baby? Does that baby have to remember this ridiculous lecture they heard that day? Not to worry. You can do whatever you like when you leave this lecture. You are affecting the expression of specific genes in specific regions of the brain. You're not producing a mutation, just altering the level at which that gene works. This does not affect the sperm. This does not affect the egg. This is selected regions that this affects. And it gives rise to the growth of new synaptic connections, which in Aplysia are quite profound. This is a sensory neuron and a motor neuron before long-term sensitization training. This is a sensory neuron and a motor neuron after sensitization training. If you actually count the number of synapses, it's more than doubling. So this is about 1,200 in a control. You go to something like 2,800 in a sensitized state. Not to worry. In your brain, you don't have growth of this size. But the interesting thing is you do have growth in your own brain. This is you. This is the way you look to yourself when nobody else is looking. This is a pre-Victorian version. I can assure you the genitalia are well represented. This is a sensory map. There's an equivalent motor map that I referred to before. This is a sensory map. When I was a medical student, I was taught that that map is fixed, that it is invariant. We now know from the work of Mike Merzenich that this is not the case. Michael looked at the representation of the fingers on the cerebral cortex. Each finger has its own little strip on the cerebral cortex. And when he examined 5 monkeys, he found that each had a somewhat different representation than the other. And he wondered, are these animals genetically different, or do they use their hands in different ways? So he had them work a bar for the only source of food that they got, pellets of food, with 3 fingertips of one hand. And he found as a result of that, that the representation of those fingertips expanded. This is really quite profound because it indicates that every single person in this audience has a slightly different brain than everybody else in this audience, if only because of the fact that they have different experiences. Identical twins with identical genes will have different brains because almost certainly they will have different life experiences, which will cause different anatomical changes in their brain. Moreover, as your mother told you, start early in life because that's when your brain is most plastic. And this is remarkably evident in the case of string instrument players. String instrument players bow with their right hand. That's like a fist. There is no differentiation of the fingers at all. And if you look at the representation of the right hand, there's no difference in string instrument players and people who don't play string instruments. But if you look at the left hand, which does the fingering, there's a remarkable difference in string instrument players compared to people who don't. And if you followed your mother's advice and started playing early before puberty, the representation is much larger than it is later on. Because one of the characteristic features of young brains is they have extraordinary capability for plastic changes, for plastic growth. Let me now turn to explicit memory storage. Ask the question, what is its nature, and how does it compare to implicit memory storage? And I should say at the outset that almost everything I'm going to say right now has really been pioneered by three Brits who are sitting here, by Tim Bliss, by Richard Mars, and by John O'Keefe. One of the things that the hippocampus specializes in is in place, and we owe that to John O'Keefe. In fact, this is work from McGuire, who is a colleague of John O'Keefe's. If you ask, this does not work in New York, so don't do this experiment in New York. If you ask a New York taxi driver to take you from Brooklyn to the Bronx, he will get lost. But if you ask a London taxi driver to take you from Hyde Park to Primrose Hill, not only will he do it, but if you ask him to just think about it, the hippocampus lights up. And that's not surprising. He has to make a conscious effort to think about it. But what it does point out, which John O'Keefe first made us aware, is that space has a particularly rich representation in the hippocampus. Not only in people, in fact, we learned that about people later on, but in mice as well. And you can study this in mice in this way, which we have used. This is developed by Carol Barnes, and we've adopted it to mice. You put the animal on a platform which has a lot of holes, and you shine light on it, and you make noise, and the animal doesn't like it. Mice do not like noise. They don't like lights. They don't like the attention. They don't like the publicity. They want to get away from it. The only way they can do that is to find the one hole out of 40 escape hatch that lets them do this. And after a while, they get very good at this, and they use the markings on the wall to guide them. They see, aha, the hole is in relationship to that strip over there, and they head directly to it. They look around, they see the strip, and boom, they go for the hole. How does this work? Information about space is mediated by a number of different modalities: visual, tactile, proprioceptive, smell. That is carried into the brain through the primary sensory areas, then to a strip of cortex called association cortex, and then it goes to the entorhinal cortex. And we now know that there is actually some sort of a very elementary spatial representation in the rhinal cortex from the work of the Moses. And that information is then carried to the hippocampus. And in the CA1 cells, we see a very specific representation of space that John O'Keefe showed in 1971, I believe. Is that right, John? God, it's a long time. Tim Bliss, who's sitting next to him, this was not planned. They just sat down like this. Discovered that if you stimulate pathways within the hippocampus, you can produce a change in synaptic strength, analogous to what we saw in Aplysia. If you give one train, you produce a transient strengthening, an early phase, but if you repeat that 4 or 5 times, you get a persistent phase. And this, like the late phase of synaptic facilitation in Aplysia, requires new protein synthesis. We have a fairly good, as a result of work of a number of people, including Richard Morris's important contribution showing how important the NMDA receptor is, we now have a fairly good understanding of how this works in molecular detail. Glutamate transmitter activates the target cell in CA1, allows calcium influx into the postsynaptic cell, activates an enzyme, a different enzyme than the cyclic AMP-dependent protein kinase, and alters the strength of this connection by altering the effectiveness of this receptor, by also inserting new receptors into that site. But if you give repeated stimulation, you recruit a dopaminergic system, and that activates a cyclic AMP pathway, and you now have a late phase that is very similar that you saw in Aplysia. MAP kinase is recruited, the cyclic AMP-dependent protein kinase is recruited, CREB2 has to be removed. CREB1, the activator, has to be activated, and you act on downstream genes, and that gives rise to what is thought to be the stable form, which also is believed to be a growth of new synaptic connections. But the evidence is less complete here. The nice thing about mice is you can exploit genetically in very elegant ways, and I'll show you one early experiment we did. We wanted to know is what happens if we knocked out the cyclic AMP system and separated the early phase from the late phase. So we generated a mouse in which we compromised the cyclic AMP system, and we could express it so it was enriched in the hippocampus. This is the dentate gyrus, CA3, CA1, and we looked at those mice. And we looked at the early phase of LTP produced by one train. This does not involve cyclic AMP, and this is perfectly normal. When we looked at the late phase, this is the normal mice, which we call wild type, but two lines which have the cyclic AMP-dependent protein kinase compromised have a clear deficit in the maintenance phase of LTP. What does this do to behavior? If we now look at animals learning this maze, wild-type mice do very well, but animals that have the cyclic AMP system compromised do very badly. So that indicated that despite the fact that these two systems, implicit and explicit, have completely different logic, use completely different neural architecture, and one is much more sophisticated than the other. Molecular biology is achieving here what is achieved in other areas of biology. It allows one to see commonalities in otherwise quite disparate phenomena. In both cases, modulatory transmitters are importantly involved. Although the short-term phases differ, the setting up of the long-term phase has a number of features in common: repressors, activators, transcription, and the growth of new synaptic connections. So with this as a background, one can begin to try to use this insight, and I'm going to end with this, to get an early understanding of diseases of memory storage. And a number of people, and John Hardy, I think had to leave, was here before, is one of the pioneers in Alzheimer's disease. This is an area where many people are making really important contributions. Let me just give you a little epidemiological background to understanding memory storage. If you take 100 people, age 70, in perfect health, no evidence of depression, no neurological disease, no cognitive deficits, and give them very, very sensitive tests for memory, you find that 40% of them, that is everybody over 70 in this audience, functions as they did in their 30s. This is called successful aging. 60%, that refers to you, madam. 60% have some sort of forgetfulness. Initially, you cannot distinguish between the two of them. But with time, you can distinguish there's one form that progresses very mildly, never becomes terribly painful. I mean, it's bothersome. People forget names, and they forget where they put their cars. But it's a very mild progression. The other 30% goes on to develop a devastating disease, which is Alzheimer's disease. We have some insight into both of them. Let me begin with benign senescent forgetfulness, the more benign disorder. Mice do not develop Alzheimer's disease spontaneously. You can put an Alzheimer's gene, a mutated gene, into a mouse and cause it to be Alzheimer's, but on their own, it just doesn't occur in mice, okay? But they undergo age-related memory loss. So if you give them this task, which you've gotten to know and love, young mice do very well, 6-month-old mice do very well, but 12 months, which is mid-age, because animals live to be 24 months, you begin to see a decline. And this is also for benign senescent forgetfulness in people. It begins not at 70, it begins at 40. And it becomes more profound at 18 months. If you now look at the hippocampus in these animals, you see that one train is completely normal, but 4 trains, these are the young mice and the old mice. The late phase is compromised. This looks amazingly similar to what you see when you compromise the cyclic AMP system. So this suggested the possibility that maybe one can reverse this by simply boosting up the cyclic AMP signaling. And you can do it in one of two ways. You can either give more of this dopaminergic input, which comes through a specific receptor called the D1, D5 receptor, or you can simply prevent cyclic AMP from being broken down. Most substances in the body are broken down, and there are specific enzymes that do this. In fact, there is a specific enzyme called the cyclic AMP-dependent phosphodiesterase, and there's a drug, Rolopram, there are lots of drugs now, that inhibit it, so it keeps the cyclic AMP level up. So there are two ways of getting up the cyclic AMP level, and we can see whether or not this is useful. In fact, we find that if you use this dopamine agonist that stimulates the dopamine receptor, you take this LTP, which is defective, and you can improve it substantially. Moreover, you take these mice that are aged and have a defect, and you give them Rolopram, you see that the behavior also improves. You bring them almost to the level of young mice. So you can get a significant restoration. So if you're a mouse, we can do a lot for you. People, one is not sure as yet. One can not only do this, one can not only do this with giving the dopamine agonist, but can also do this by giving Rolopram. And let me simply show you that you can reverse the defect with Rolopram as well. So that brings me to the final point. What about Alzheimer's disease? Well, I must say one of the really remarkable things about Alzheimer's disease, since John Hartley's discovery that the critical defect is a metabolism of a particular protein called beta amyloid, how much of an understanding we've gained in Alzheimer's disease. And there are a lot of strategies now being used to try to combat it. I'm only going to describe you one set of experiments. One can present 10 alternative ones. So this is only one way of looking at it. This is not my own work. This is work done at Columbia, but had nothing to do with me, by a guy called Mike Shalansky. He made an interesting observation. He saw that in patients with Alzheimer's disease, you can see significant cognitive impairment before you see the characteristic plaques that define Alzheimer's disease. Before beta amyloid sort of crystallizes out and forms extracellular junk, it already does damage. And he showed that if you just take a solution of beta amyloid and put it onto brain tissue, it causes toxicity. It's bad for cells. And when he explored these cells, just pouring on beta amyloid, he found to his astonishment that what it did was to activate inhibitory constraints that dampen down the cyclic AMP system. So it activates a phosphatase, it activates CREB2, the repressor of CREB1, so it prevents the cyclic AMP system from working. So it dawned on him that maybe here also a drug like Rolopram might be useful. So he began to look at a number of indices. One, that CREB, the transcription factor, is dramatically decreased in its level of phosphorylation, its level of activation in mice in which the beta amyloid gene, the mutated gene, has been expressed in mice that develop Alzheimer's disease. But if you give Rolopram, you can restore in good measure the degree of phosphorylation. These mice show a memory deficit. So you're putting a beta amyloid mutation into a mouse. They show a significant memory loss on this particular task. Again, Rolopram restores that. But the most interesting thing is you also have severe anatomical damage. You have retraction of spines. And again, this spine retraction is returned by Rolopram. I do not want to give you the indication that this is a magic bullet. It is not. I'm just indicating to you that there are now a family of approaches that are being used. I gave you Rolopram because it made it easier to follow the argument I was developing earlier. This is something that overcomes the damage due to beta amyloid. Other strategies are designed to reduce the toxicity from beta amyloid by decreasing the load, either breaking down the abnormal product more rapidly or preventing the buildup of the product. So even though at the moment there is no satisfactory treatment for Alzheimer's disease, I'm confident that with this massive approach of biology that we should have something which at least is moderately useful within the next 5 years. So let me simply end by saying that one of the wonderful things of biology is not only that it provides insight into living processes, and biology of the mind, of course, gives you insight into how we function as human beings. But also the great hope of biology of the brain is that these devastating diseases of the brain, of which Alzheimer's and benign senescent forgetfulness are just two examples, but schizophrenia and depression, anxiety states, all of these things we're beginning gradually, in some cases in very primitive ways, to understand at molecular level, and that hopefully in this next century we will be able to use this molecular biology in order to be useful to treating people who suffer from these diseases. Let me simply end by thanking the colleagues who did the work in my lab. Yanyu Wang, Ted Abel, Mary Elizabeth Bach were involved in generating the mice that I showed you. Mark Barad, Vusika Bulchinazzi, Gail Malloret, and Minzo were involved in the animal models. And work that I was not involved with was Michael Shalansky and his colleague Octavia Rancho. Thank you very much.
We're running a bit late. Can we have two questions?
Absolutely.
Okay.
Whatever you like.
Okay. We're running a bit late, but I think we should have 5 minutes for questions. If you want to ask a question, please put your hand up and wait till the microphone reaches you. So who would like to start? Yes. Question there.
Would you agree that long-term memory is better served if it's accompanied by a strong emotional stimulant, i.e. trauma? There is no need for repetition.
Oh, absolutely right. I mean, I skipped over it a little bit rapidly, but I did emphasize that in some circumstances you can get one-trial learning. And emotion, of course, is a perfect example. If, you know, in America, the typical thing was the Kennedy assassination or Martin Luther King assassination. People know where they were at that particular moment, even though it was a single experience. And one of the reasons one sees this tremendous increase of post-traumatic stress disorder, many of these, a person being physically assaulted, a woman being raped, these are things that even though, you know, it's a transient exposure, can lead to very persistent memories. Absolutely.
Hi, thanks for the talk. I'm a philosopher, so I don't know that much about the neuroscience, but I'm very interested by the distinction between explicit and implicit memory. So you gave this example of the taxi driver rehearsing the route he would take from Hyde Park to Primrose Hill. And that was an example of explicit memory. He would consciously rehearse the route, right? But it struck me that my dad's a black cab driver, so I have some acquaintance with this phenomenon. But he would more typically just be talking your ear off while he drove his way on that route without actually having to consciously entertain the route. Wait, so that looks more like a case of implicit or procedural memory. So it made me wonder: is the same information available to both processes, to both systems, or is it not the case that there's such a clear factual, non-factual distinction between them, or how would you pass that?
That's quite brilliant. That's exactly the way it works. Many things that start out as explicit memory, de Kooning's paintings, become implicit after a while because it's so practiced. So it's different if you ask him, think of the route, okay? He has to think. The other thing is he's actually driving, you see. So those are really two different situations, even though the same spatial geometry is involved. So you're absolutely right. Many things, learning how to ride a bicycle, many things that begin explicitly, once they become practiced, become implicit. And it's a way to take the functional strain off the explicit system.
Thank you very much for this fascinating talk. Is there any way of knowing if you or somebody else has got the benign senescent forgetfulness or the mild cognitive impairment?
There, in principle, should be. People are developing methods, and I'll give you a very simple answer. It is not absolutely trustworthy at this point. Alzheimer's disease begins in the entorhinal cortex. You can see this in pathology. That's where the plaques are first deposited. And with imaging experiments, people see abnormalities in the entorhinal cortex in early Alzheimer's disease. Benign senescent forgetfulness does not begin in the entorhinal cortex. It is evident in the hippocampus. The evidence for that is weaker. It's just beginning to emerge. But there is beginning evidence that there are two different, both of them involve the medial temporal lobe. In fact, both of them begin by involving a defect in explicit memory storage. I sort of implied that with the de Kooning example. He couldn't recognize anybody around him, but he could still do. And patients, people, normal people with age-related memory loss, it's primarily explicit memory that is defective. Jim Watson tells me his backhand is getting stronger all the time. He's 80 years old. So, in fact, you see this in terms of skills, that you can continue to improve your skills. Oliver Sacks actually was telling me the other day that he's working to improve his implicit memory. He's going back and taking piano lessons so he can make sure that those skills will stay with him if, God forbid, he should ever develop Alzheimer's disease.
Front here, yes.
Could you give me some ideas that I could have a successful life-term memory?
I cannot guarantee it, but let me tell you what the current thinking is. The current thinking is, again, your mother probably told you, a healthy body makes a healthy mind. So good physical health, activity, low blood lipids is a protective factor for cognitive decline. Issue number one. Issue number two, for which the evidence is less good, but intuitively many of us believe it, being intellectually active. Doing things that put a stress on your cognitive capabilities. That is not necessarily doing what you do routinely. That is, if you're, I don't know, a lawyer or a physician, practicing your trade, even though it may be highly intellectual, probably to you is largely implicit at this point. So it would be good for you to do something which really, learning a foreign language, learning how to play the piano. This is, at the moment, on the level of folk medicine, but this is the current teaching. And there are programs that are coming on the market that are designed to sort of exercise your mind. There are no really controlled studies to show that they work, but I think it's likely that they're going to be useful.
Two more questions. One there.
I think you said you'd been doing research in this field for about 5 decades. And so if you were thinking ahead several decades, could you talk about some of the known unknowns or unknown unknowns that, going back to your political thing,
I do think that.
With regard to memory, what could you imagine could be some of the things you might be there to discover?
There are enormous problems that remain, okay? So we know, for example, that John O'Keefe, in the classic set of studies, showed how different cells in CA1 respond to different positions in space as the animal walks around. How that information comes in and gives rise to the place cells he described, we did not know until recently. Now the Moses have shown that there are cells called grid cells in the entorhinal cortex that map the external world in a rather fixed way. To learn how that information from the entorhinal cortex gets transformed to give rise to John O'Keefe's place cells is a fantastic problem, which very likely is going to be solved in the next 5 or 10 years. Where the information goes outside the hippocampus, we don't know. So I told you that HM has perfectly good memory for things that occurred prior to the operation. And you can show this in a mouse. You can teach a mouse, for example, a task and lesion the hippocampus. If you lesion the hippocampus a day after you've taught the animal an explicit task, it has no memory whatsoever. If you lesion the hippocampus a week or 2 later, it has a little bit of a memory. If you lesion it a month or 2 later, it's as if you didn't do anything the information is already moved to some other site and it's perfectly well stored there. So you can do without, in the mouse, you can do without the hippocampus 2 or 3 months after you've learned something. Where is that other site? We think that what happens is when I look at you, the image that you cast on my retina goes to a part of the visual system which is concerned with the recognition of faces. You put an electrode into that region, 98% of the cells respond only to faces. You make a lesion in that area, you get prosagnosia, a selective defect for face recognition. So we think that your face makes an imprint on the area involved in face recognition, but that imprint is weak. It will not carry the memory by itself. And somehow, in ways we don't understand, but one would like to solve in the next 10 or 15 years, the hippocampus reinforces that initial imprint and makes it stronger and stronger over a period of time. That's a wonderful problem. It's largely a mystery. There are many mysteries like this. I'll end up by giving you, not the mystery, a thing that bothers me. I'm incompetent, but I'm trained as a psychiatrist. And it really strikes me that in the last 20 years, we've had zero improvement in the drugs that treat schizophrenia and depression, the major mental illnesses. In fact, recent studies showed that chlorpromazine, which was available when I was a resident in 1960, even though it has more side effects, is as effective in its curative powers as anything that has come since then, the atypical antipsychotics. The selective serotonin uptake inhibitors that were developed 25 years ago are as good as anything that has come along since then. We have no new insights that have led to new therapies in the most important mental illnesses. In fact, the astonishing thing in this era of molecular biology, the one thing that has become more supported by scientific evidence is that we now have modes of psychotherapy that have been validated in controlled clinical trials. Cognitive behavior therapy, which is extensively used in England by the Health Service, which Aaron Beck developed, has been shown to be extremely effective, as good, if not better in certain cases, for depression and selective serotonin uptake inhibitors. So this is a behavioral insight. The molecular biology so far has not given us what we needed to give us, is new treatments for these major disorders, and hopefully that will come. But listen, I'm at an early point in my career. I really look forward to having a chance to crack some of these other problems.
I think we could go on a very long time, but probably ought to stop the questions now. It's clear that understanding the brain is the Mount Everest of science, compared to which the physics and astronomy some of us do is a complete doddle.
Not true. Some of my best friends are astronomers.
Right. But it's equally clear that we couldn't have found a more engaging, eloquent, or authoritative guide to what are still the foothills of that subject than Eric. And so I'd like to ask you to join me in once again thanking him for a really wonderful lecture.